Morning, afternoon. It's just 12 o'clock, so however you want to have the cutoff. My name is Amy Wagner-Johnson. I'm in the EIRH and the REPE themes, and it's my pleasure to introduce Dr. Christina Laukaitis. She's an associate professor in clinical genetics, so that's in the College of Medicine here. But she got her MD, PhD here at University of Illinois in the Legacy School. She did her residency in internal medicine at St. Vincent Hospital in Indianapolis, and a medical genetics fellowship at the University of Washington. She is board certified in internal medicine and medical genetics, and she joined the faculty at the University of Arizona College of Medicine in 2008, and then moved here in 2001, so we're really excited to have her here. Her clinical practice and research focus on diagnosing and managing patients with genetic syndromes. She's published her work on caring for people with hereditary cancer syndromes, lysosomal storage diseases, and connective tissue disorders. So today she's going to talk, I think, primarily about EDS, and I will let her take the floor. Thank you.
Thank you very much. It's wonderful to be here, and it's wonderful to be here in person, where I'm not speaking to a microphone and a camera, and there's actual people in 3D in the room. So I'm going to be talking about hypermobile EDS, which, as it says, is a very complicated, poorly understood connective tissue disorder. And so I'm going to be talking about my group's research trying to understand the etiology of this condition. Okay. So EDS or Ehlers-Danlos syndrome, as an entity, was first recognized around the turn of the last century by a couple of dermatologists who kept seeing patients who had unusual skin that bruised easily and was sometimes very loose, and they noticed that many of these people also had joints that were extra flexible, moved beyond the typical range of motion. And so Dr. Ehlers and Dr. Danlos named this syndrome after themselves, and we have been trying to understand it ever since. So in the last 120 years, we've understood slices of this condition and now recognize many subtypes. What's left is a large chunk of patients who we don't have a clear etiology or cause or treatment for. And so that's where my work comes in. One of the characteristics of many connective tissue disorders, including Ehlers-Danlos syndrome, EDS, is joint hypermobility. And this is a schematic of what we use, one scale we use to measure joint hypermobility called the Baton score. So this is a way of asking, do multiple joints in an individual move beyond a typical range of motion? So if everyone takes a minute and does their Baton score together, based on bilateral symmetry, a person can have a total Baton score of 9, where you'd get 2 points for each pinky, 2 for each thumb, 2 for each elbow, 2 for each knee, and then 1 for touching the ground. Technically, this is normal. This isn't quite as far extended as it should be. It should be 90-degree angle between the bones of the carpal bones and then the finger bones. So that if a score, depending on age, of 5 or above is generally abnormal and indicates generalized hypermobility. It's normal to have 1 or 2 joints that are positive per this score. It is, it would not be typical for every joint to be hypermobile, and that makes us look for other issues. I should also say 3 other things about the Baton score. Number 1 is we know that the score will change by age. Younger people tend to be more flexible. As people age, they become less flexible. There are gender influences on the score. Males tend to be less flexible than females, and muscle tone makes a difference. And number 3, this is really easy to do. It's easy to teach. It's easy to do in clinic, and it doesn't capture many of the most relevant joints, the joints where people have the most issues, like shoulders and hips and ankles. But it is a score, and it's what we use. Okay. So, as I said, over the last 120 years, we've recognized a number of different specific types of EDS. Some of these became recognized because people have some remarkable features, like very stretchy skin, which is characteristic of classical EDS. So skin stretch at the volar forearm of more than 3 centimeters is where I start thinking we have a classical EDS. Kyphoscoliotic EDS, where people have very severe kyphoscoliosis, often to the degree that their stature is affected and surgery is almost all universally required. The newest type of EDS to be recognized is periodontal EDS. Here, the gingiva and the bone do not connect, and so people will have tooth loss by their teens, in addition to other signs of connective tissue problems. So it's beyond what is often seen in weak teeth or periodontal disease, but a very severe specific phenotype. Most of the genes associated with these subtypes have to do with collagen or extracellular matrix structure. Classical EDS is related to mutations in collagen 5A1 and 5A2. Vascular EDS is related to collagen 3A1 mutations. Collagen 12, collagen 1 are also important in other types of EDS. Some of the rare types of EDS are related to mutations in genes encoding for enzymes like lysyl hydroxylase, which is involved in collagen cross-linking. Others are involved with or cause dysregulation or dysfunction of enzymes that make extracellular matrix molecules like glycosaminoglycans. A few are in transcription factors and other matrix metalloproteases. And then periodontal EDS, the most recent one to be linked, is actually caused by mutations in complement genes. So it's not just about the tissue, it's also about our response to tissue. So complement mutations are one of the more interesting links that we see. You know, how is it that people with periodontal EDS who look just about like vascular EDS, except they also have teeth, actually have an immune system dysregulation? It's an interesting mechanistic question that we'll come back to. But the biggest issue in this set of conditions is that the hypermobile EDS type doesn't have any gene associated, and that causes a variety of issues, both diagnostically and from a treatment point of view. So what is hypermobile EDS? Well, we know it's common. When I talk about hypermobility, I like to remind people that just being flexible is not a disease. Depending on age and ethnicity, if you go into a classroom of children and check the Baton score, upwards of 20% of children might have hypermobility. And all of these children don't have EDS and don't have a disease. So just being flexible predisposes people to problems, but doesn't guarantee that everyone will develop medical issues. So, you know, as a general rule, maybe 1 in 4 people would have generalized hypermobility. But a much smaller percentage of those people are going to actually have a syndrome or a diagnosis. And so for me, the threshold for diagnosis is, does this hypermobility relate to, or is it the best explanation for other medical problems, chronic pain, chronic joint instability, et cetera. And so when you use that kind of definition, about 1 in 100 people has or could be diagnosed with a hypermobility-related syndrome. And maybe 1 in 500 people with hypermobile EDS. And I'll go over the difference between these two very closely related diagnoses. This all relates to the much more rare other EDS types, those other 13 types that have genes associated, where as a whole, about 1 in 5,000 people will have one of those 13 very rare subtypes. So hypermobile EDS is 10 times more common than all of the other types of EDS combined. And it's even less common than people who also fall on this spectrum of having hypermobility and functional issues. And then genetics gets involved not only because, you know, we know that some of these types of EDS are clearly genetic, but also because there's many other genetic syndromes where hypermobility is a feature. Things from Fragile X to chromosomal deletion syndromes to Noonan syndrome. So when I'm seeing a patient, you know, my job here is to make sure this isn't something else. So in addition to being much more common than one might think, EDS is also challenging. These are data from one of our studies in Arizona, where we asked our 150 participants about their health. So we said, how do you function? How is your general health? How is your physical and social functioning? How well are you able to perform your roles, your physical roles at work and at home, your emotional roles because of your health? And here on the SF-36, which is a score of that, higher numbers mean a person is functioning better. Lower numbers mean a person is functioning worse. So these yellow and orange bars are population normals and unaffected people from some of our studies. And people do pretty well. A little bit of variation in vitality, but, you know, pretty well. But people with in the yellow or green lines are people with hypermobility spectrum disorders or hypermobility EDS. And nearly universally, they have challenges in multiple areas of physical and emotional functioning. So this highly impacts quality of life of individuals with these diagnoses. There are some interesting differences in emotional roles between people with hEDS and GHSD that we think need more evaluation. Are they something specific to that population, or do they indicate some areas where we could do psychiatric or psychological intervention to help people function better? But third thing that's remarkable about EDS is it's poorly understood, partly because it lacks a biomarker. We don't, the best diagnostic strategy we have for hypermobile EDS is to send them to genetics and make people wait 3 years to get in, and we do a clinical exam. That's kind of what we got. Thankfully, there's been some press recently. There was a big CNN article just came out over Christmas talking about raising awareness of EDS. There's been a little bit more edgy article in Stat talking about gaslit patients who are now being trained as scientists to pursue questions about EDS. So awareness is out there, but without a biomarker, it remains a challenging diagnosis. So this, as I say, is the central core of my group's work. What is causing EDS, hypermobile type, and how do we help use that information to help people have an improvement in quality of life? The questions are, why haven't we found a gene? People have worked for 120 years, and we've come up with, 24 genes for other types. So why has this not worked in hypermobile EDS? One hypothesis has been that the cohorts of people we've been studying for gene finding are just too heterogeneous. This makes some sense. If you think that we've taken a very mixed pool of people and we've pulled out in slices 13 other types that link to a specific gene and have a fairly homogeneous phenotype, then the people that are left are going to be pretty mixed. So rather than looking like this upper quadrant where we have a beautiful case control studies where our cases and our controls separate along different axes and are clearly differentiable, we're probably more like what we're seeing in these other 3 quadrants, where cases and controls might be mixed, the same genetic factors might in different amounts be causing cases and controls, and we might have multiple different causes in our cases. So all 3 of these probably play some role in what our challenges are. It might also be that we're looking in the wrong place. People have tried for decades to understand what causes autism and schizophrenia, other common diseases that run in families, and it wasn't until we stopped looking at single nucleotide variation and started looking at copy number variation that we started to get a toehold into those diseases. Maybe EDS isn't a point mutation in collagen. Maybe hypermobile EDS isn't from a point mutation in collagen. Maybe it's from something more complicated. We know there's lots of duplicated gene families like the cytochromes, the immunoglobulins, some of the serine proteases that are hard to evaluate even with very good whole exome or whole genome sequencing. This kind of gene family doesn't do well when you're doing next generation short read sequencing. So these are going to be not studied by current techniques. There's regions of the gene that are copy number variant. These 2 overlap. There's also lots of non-coding sequence and questions about mitochondria. So we need to broaden where we're looking. You can't keep doing the same experiment if it hasn't worked for 120 years. It's possible that EDS has some polygenic components. Some of the other types of EDS fall clearly into this Mendelian area. We have very rare mutations with very high effect sizes. If you have the mutation, the probability that you will show the phenotype is very high. But many common diseases are related to more common variants in the population that have lower effect sizes. Having a single risk allele might not be enough to cause disease. You might have to have 2 or 3 risk alleles in order to have disease. Again, this is where GWAS comes in, and you need bigger population cohorts. Finally, we might be dealing with a complex disease in the truest sense, where we've got both genetic and environmental components. And so it might be that a person comes to diagnosis because they have a number of risk alleles, and there's been an environmental insult, a viral infection, a bad car accident, early life trauma. And those things add together to lead to this constellation of symptoms that get someone past their primary care and into genetics. So again, this is tougher to tease out genetically with sequencing, but is part of the equation that needs to be taken into account. So So this is a good time then to talk about this difference between hypermobility syndromes and hypermobility EDS, because I think this helps to understand both where my research has come in and how we're addressing this issue as a greater community. Prior to 2017, if someone had generalized hypermobility and chronic unexplained pain, they were given a diagnosis of EDS type 3, which was the hypermobility type EDS. But again, we've failed to identify genes associated with that. And so one of the reasons that the thinking goes that is we had complex patient groups that were not terribly homogeneous, and we needed to try to tighten the box for gene finding to make a smaller group of people who got in there for, the most biological reasons that we could come up with, the least environmental influence. So we made some new criteria. And so since 2017, we've been using these new criteria to define hypermobile EDS. And these are simple, right? Everyone's ready to go home and do them? Basically, I'm not going to go into detail. If you would like a diagnostic session someday in your clinical world, let me know. But for most of us, the points are a person to be diagnosed with hEDS has to be hypermobile. They have to have a Baton score, generally a 5 or above, depending on their age. And then these new criteria focus on objective signs, things that are not symptom-based but are observable and measurable. So I'll go over those. We still take into account, like many other genetic diseases, if someone has a family history, then the burden to get above the bar to meet diagnosis is lower. And we do take into account pain and joint instability, those signs of a syndrome. And then finally, criterion 3, this bottom bunch of words are my job. It's to rule out everything else that can cause this combination of things. So what are these objective signs? These are what I would call or categorize as connective tissue signs. These are signs that your tissue is made just a little bit differently. They have to do with soft, velvety skin hyperextensibility, unexplained stretch marks, physiogenic papules at the back of the heel, hernias that are recurrent, scarring that's unusual and atrophic, pelvic floor prolapse. So rectal prolapse in someone who's never had children is a big red sign, a big red flag that tissue is made differently. Some of these are growth. They have to do with dental crowding and high-arched palate, arachnodactyly, so long, thin fingers, high arm span to height ratios, people who look somewhat Marfanoid. Sometimes that's a sign that TGF-beta is upregulated in some of the connective tissue pathways. And then cardiac issues. If there's mild aortic root dilation or mitral valve prolapse, those are supportive, although frankly those are also red flags for me. If someone has too many of these, well, they might well meet the EDS criteria. That's when I'm wondering about another connective tissue disease. Like I say, these are fairly objective, but some of them not so much. Soft velvety skin, for example. Try teaching that to someone. It's impossible and it's hard to define it. So you just have to hopefully have seen someone with it and then feel it, have that vision. So we can't quantitate that one, but we can do others. Mild skin hyperextensibility means 1.5 centimeters at the volar forearm, etc. So to summarize the summary, hypermobility is a hallmark of connective tissue disease, and the most common connective tissue disease by far is hypermobile EDS. Unfortunately, it's not been linked to a single gene. It disproportionately affects women, which I didn't touch on, but we'll come back to. It significantly affects quality of life, and it's poorly understood. We've developed some new diagnostic criteria to try to increase the objectivity of diagnosis, but really they're most useful in the research area, which is in recruiting homogeneous research cohorts. So I'm going to talk about my group's work in 3 areas. People will recognize our central dogma of DNA, RNA, and protein, and I haven't strayed very far from that in my work. So we'll talk about our work in DNA, our work doing some omics studies, which involve both RNA and metabolomics, and then if I have a little time left, I'll tell you about our phenotypic descriptions. So in about 2017, Peter Byers, my old fellowship advisor from UW, and I developed what was called the hEDS gene study. It was a linkage-based trial to try to identify genes associated with hEDS. We took advantage of these new criteria and used them extensively to recruit our cohort. So we were targeting people over the age of 18, doing physical exams, making family histories, collecting biosamples, and asking them to each do a set of questionnaires. We were hoping to get 150 people, and we did. We got about 156. We were hoping to only get people with EDS, hEDS, and informative negatives from their family. Because of the way recruitment developed, we did get a small cohort who had hypermobility spectrum disorder, who were kind of in that 1 in 100 group. It turned out that was useful. It allowed us to do some symptom comparison and some testing of the new criteria. So at the end, I can come back to that. We ended up getting people from 38 families, 150 people from them. We got a few really nice big families with third-degree relatives. We got one set of families, or one family were third cousins, so seventh-degree relatives. Thank you, Utah. We also got a bunch of small families. 26 of our families had multiple first-degree relatives. So first-degree relatives are people who are siblings or parent children. Those are useful, not quite as powerful genetically as these big extended families, but useful. And then we had 41 individuals that we're using for follow-up. So we'll do the linkage on these 38 families and then follow up and see who's in the individuals in the cohort. The cohort is described here, and what's remarkable is it's female. We didn't try to recruit based on gender, but ultimately 90% of our people with hEDS and 80% with HSD are female. That's as opposed to about half of our unaffected. So in the literature, we usually read 2-to-1 female to male predominance. In my clinic, in our studies, it's more like 10-to-1. So we can argue over why that is. I'm not, but it is. Obviously, people who met criteria were more flexible, had many objective signs, and I'd just like to highlight the pain. 90% of these individuals in the cohort have chronic musculoskeletal pain of unexplained origins. So, again, this is a quality of life issue. So we are in the process of completing our evaluation of 48 exomes and 79 genomes, and so I'll kind of give you the status on that, and then we've completed some other phenotypic descriptions of the population. The genetics work was started by Kiana Martinez, who's since graduated, and we're finishing up replicating and getting ready to hopefully get a story pulled together. So the genetic landscape of hEDS is we know about 3 candidate genes. One is the gene encoding tryptase, which is important, we think, in histamine regulation. The second is tenascin XB, which encodes a protein which is a linker in the extracellular matrix. It's one of the proteins that links collagen to the other ECM proteins. And the third one is something in the 8p21 to 8p22 region that links in one Belgian family. So, that's kind of what we know. What is interesting about 2 of these is that these are point mutations play a role here, but for the most part, these are structural. They're triplications and rearrangements. And the TNXB is the one that's the most intriguing and interesting. So just to elaborate on the story, one of the rare types of EDS, classic-like EDS, is an autosomal recessive EDS related to biallelic mutations that eliminate function of both copies of TNXB. And this is often actually in conjunction with a second genetic deletion in the area. The TNXB gene is right downstream or upstream, whichever, of CYP21A2, a gene that's important in the other genetic disease, congenital adrenal hyperplasia. So CAH families, if they're happening because of biallelic rearrangements, will often have significant hypermobility and the recessive type of EDS. The carrier relatives in those families, the families who are carriers and have a single elimination of a single copy of TNXB, have hypermobility if they're female, but not if they're male. At least that's what the data show. 0% of male, 60% of female heterozygotes from classic-like EDS families had hypermobility. And so based on these data, 20 years ago, the Dutch group who was working on it went to a Dutch EDS convention and assayed everyone there with EDS3, which is the old, slightly bigger mixed bag. And they found that indeed about 2.5% of that cohort had TNXB rearrangements, but about 7.5% had low tenascin X protein levels. So at that point, work on this locus kind of faltered because it's really hard to sequence. It is a rearrangement that's a nightmare to identify. You're not going to find it on your exomes. And the assay for it is fairly challenging. You're trying to sequence through a large duplicated region, and you have to use a long-range PCR, like a Tacrolaw PCR, to make a 30 kb piece that has anchored outside the duplicated region and then sequenced through manually. So it's a bit of a mess. So because the technology wasn't there and it didn't seem to explain much of hypermobile EDS, no one has done much more with it. But the region really is interesting because beyond tenascin X and CYP21A2, it also includes a complement gene, complement 4. So, and this complement 4 gene has actually been studied a little bit more extensively in the schizophrenia literature. So there's a number of groups who are interested in the connection between this complement gene and a number of mental illnesses. And so there's so many intriguing reasons and connections why this is an interesting candidate gene that my group has focused on it. If you've known any of my other work, you know that I tend to like complicated gene families, things that are challenging and difficult and can't be done in an automated way. So it matches. This is where I belong. So we went through our short reads, knowing that these were not going to be full assays, but that they might give a hint of, is there a problem? Could there be an issue? And indeed, we found that there's areas where there are differences in the number of reads within this tenascin X region. So this is plotting where the 30 kb deletion is, and some people have less reads in that area. This is not causative, it's not proof, but it could be a fingerprint of issues. So we went through earlier on and looked in our population, and of the 19 families we had good data on had genome data on at that point. We found about 3 that looked like they had that fingerprint of deletion, about 13 that looked like they might actually have duplications or more complicated duplications deletions, which is a phenotype that's seen, the trimodular, and about 3 had nothing. So it looks like there's a lot going on. In the individuals, again, a lot had rearrangements, some didn't. The challenge is, because this is poorly sequenced and poorly characterized, we don't have population data. I don't know whether it's normal to have, what's the normal allele frequency in a population. So, again, we kind of floundered and left it at this point because we couldn't do good comparisons to know if we had a signature of issues or not, and we didn't know what the population level should look like. We did do, not before I had one of my crackerjack young undergrads do some protein work. So we were looking at tenascin X protein levels with ELISA in serum from our population. And we again said, what's up with this? hEDS patients look like they have higher tenascin X levels than our controls. What's going on here? And again, kind of let it sit. But I'm going to come back to it because this is becoming even more interesting. Obviously, when you have 100 genomes and exomes, you also want to look at single nucleotide variants. So these are, a summary of what we found in the 5 million variants in our whole genome data. If you run, we are using an analysis called rare variant sharing, which has been used very effectively by groups looking at other complex traits like cleft palate to identify candidate genes. So we thought this would work appropriately here. We found that at the 0.001 level, we found about 1,000 candidate variants that would need more focus. As we moved up or down in p-value, that narrowed down. So we're in the process of sorting through these, one by one, to figure out which ones are worth trying to develop or seek partners for validation. And so we're following up with these in a variety of ways. So what we also found are a lot of interesting variants in collagens. So if you look at people with unusual connective tissue, they have a lot of weird spelling of collagen. So this is data from a colleague, but it's just like mine, where you can look at collagen 3, collagen 5, collagen 6, collagen 1, and there's a lot of variants in those genes that are not the type you'd expect to cause classical EDS. They're not the glycine substitutions in the regions you'd expect, but they're interesting non-synonymous variants that change the structure. And so the question is, do these subtle variations cause subtle variations? do the subtle protein differences cause hypermobility that doesn't quite reach the threshold of the skin stretchiness we'd see in classical EDS. So we're quite interested in developing assays to understand the effects of subtle differences in collagens, and I'm thrilled to have some local colleagues who share that interest. So that's where, as far as we got with the story, when an international group that came in. And so the Ehlers-Danlos Society, which is an international partnership of patients and physicians and scientists, was interested in doing a study similar to mine. And so I was on the board that helped to design this, and we used some of the lessons we learned to adjust how this was done. So the goal of what we called the hEDS trial is to do a cohort-only enrollment, look only for people with hypermobility EDS, and to sequence and do whole genome sequencing of 1,000 people with hypermobile EDS. The idea here is to, again, work with the numbers. If we're dealing with a complex disease, you have to have more people in order to map. And so we're trying to get more people. And then the idea is to use population controls as our controls. So at this point, thankfully, 1,022 people have been recruited and sequenced, and those data came off the sequencers at the Broad in September. We've now completed the PCA for ancestry, so this is telling us what the ancestry is of the population. Obviously, we can compare that to self-reported, but we'll use this for deciding controls. And thankfully, all of us, which is a large national sequencing program, was sequenced also at the Broad using the same technology at about the same time and is publicly available. So we're going to be doing 5-to-1 matching using Ancestry to get our controls for hEDS. And so that is going on as we speak. So Vasek is working on it from what I know right now. And then we're also working on curating the phenotype data. So what we're able to do now is to duplicate a lot of the analyses we're working on. We started with hEDS in a much larger cohort with a very good set of matched controls and with some great partners. So the group working on this, we have here at the University of Illinois and at Carle, we've got the candidate gene and some of the association analysis, the phenotype association analysis contract. So Vasek is my analyst, Deepak's working on phenotype, and then me leading the charge. And so we're working on, like I say, candidate genes, making sure no one in this group has vascular or classical EDS, and as candidate genes come through, testing to see if anyone has them. I'm most excited to be working with Bob Hansicker and my RN PhD student in Tucson, Maureen Galindo, to be looking at the copy number and structural variant analysis. Bob is an analyst at the Broad who has a strength in complex gene regions. He's worked with the C4 region for a decade, and he's got techniques to look at short reads and sort through the structural variations that we're expecting and that we're seeing in TNXB. We've also got, because all of us has done a subset with long reads, we're able to validate things with long reads and see if what we see on the short read data is truly a signature of reality. So we're making fabulous progress looking at TNXB and looking at tryptase. And then Joel Hirschhorn from Boston Children's is leading the group doing GWAS and burden and polygenic gene analyses. So we have 2 years of funding, and we will let you know what we see in the next 2 years. This should be exciting. So I'm going to move to protein, or to RNA. And part of our issue with RNA was we had so many candidate genes, we were struggling to narrow down which ones are relevant. We needed to figure out which pathways were relevant. So we answered a call through the international group for some skin sequencing studies. And we started what we call the hEDS-omics trial. The hEDS-omics study is a cohort of 45 people with hEDS and 45 age-matched controls, all of whom are female. We said, get that variable out of the way. 90% of people are female. We're just going to focus on the women. We collected both in Tucson and up here in Champaign-Urbana, and collected both biosamples as well as clinical information like family history and medications. So there's a lot of overlap between people in hEDS gene and hEDS-omics, but we've got a lovely cohort now. And so the samples we collected included 2 skin biopsies, or a skin biopsy we actually split in half of which we took RNA from directly for RNA sequencing, half of which we cultured before taking RNA for RNA sequencing. We took blood for serum for metabolomics, and then we have urine for hormone studies. Part of that blood we did platelet functional studies, and then we frozen aliquots of everything. And so again, this is just our local, the people we recruited up here from Champaign, Urbana. We had 60 of our recruitees from up here, about half with hEDS, half without. The age was in the mid-30s, was our median age. Baton score, as you'd expect. People with hEDS are more flexible than people without. And again, about 86% of our people had widespread chronic pain. And our evaluation strategy then is this. We're using a 4x4 evaluation strategy where we're going to be comparing between controls and people with hEDS, both at the skin biopsy level and at the cultured fibroblast level. So is what we see in skin reflect cultured fibroblasts? And how does control compare to hEDS? The thinking here is that we know that not only are the growth factors different in a plate than in the body, so are the mechanical and tensile forces. So we can, but you only get one shot at what you get from a skin biopsy. And whereas cultured fibroblasts you can get a number of passages on and do other experiments with. So we'll also then be comparing the other direction and asking how does the process of culturing simplify or change what we're seeing in the controls. So 4x4 comparison. I have some preliminary data on 24 individuals, 12 controls and 12 people with hEDS that we did RNA sequencing on this summer. And so I'll just give you a sneak peek into that. Everything else is, again, on the sequencer. Expect it back around the end of the month, maybe beginning of next. And so all of this was done with the help of our Roy Carver Biotechnology Center, who've been very effective. And so I've been I'm grateful for them for their help. So we did a PCA of our samples, and indeed we're happy to see that we could separate our samples from our controls. That's always a good thing. And then when we did that and then looked, we were able to find a number of up and down regulated genes. So each of the samples is one of the columns. The right columns are our affected people, and the left columns are our controls. Each of these dark lines in the dendrogram on the left is a gene. So 3,800 genes were either up or down regulated with a p-value of less than 0.5. It's not the strongest p-value ever, but that's why we're expanding and repeating. And so one story I'll tell from this is in the data, the other group that's done RNA work with hypermobile EDS looked at cells that they had cultured. They regrew the cells and then looked at the phenotype. And what they got was a near immediate differentiation from normal fibroblasts to fibroblasts that had differentiated into myofibroblasts, which is a more scar-type, firm type of cell. In the process, they upregulated their alpha V beta 3 integrin. And so with any of the hypermobility phenotypes, vascular, classical, hEDS, and HSDs, we see strong increases in alpha V beta 3 expression in their fibroblasts. So we're interested to see, does that happen in our cells, or at least in our skin? And so here are a volcano plot of our RNA data from our population. Those that are red, genes that are in red are those that have statistically significant upregulation in hEDS, and in blue, statistically significant downregulation in EDS. And the two spots here are alpha V and beta 3. Alpha V is actually significantly downregulated in the skin of people with hypermobile EDS compared to the controls. So absolutely the opposite of what the Italian group had seen in their cultured fibroblasts. And although these are the raw data, and those curves are not particularly far apart. So what we're seeing is that human, and then the beta 3 is this gray spot there, so no significant difference. So in human skin from people with hEDS, we don't see the same effects that the Italian group saw in cultured fibroblast of people with hEDS. Just an interesting little, beginning finding, but it's kind of a fun question to ask. We do, in conjunction with this, actually see a lot of downregulation of genes that are packaged into extra exosomes and membrane-bound organelles and extracellular organelles. So again, not quite what I'd expect. Why are we seeing downregulation? I'm not certain, but these are pretty strong, enrichment p-values when you do the pathway analyses. So from here, we're really excited to have all of our other samples come off of the sequencer. Got 72 more skin biopsy samples coming off the sequencer, and then all 96 cultured skin samples coming off. So, so these should, as I say, be off soon, and we'll be spending a spring deep into data analysis. We've also Maureen is quite interested in single cell questions, so we've got some funding through a private donor and have thrown 5 of our Arizona samples on for single cell sequencing. So I think those went on 2 weeks ago, so we should have those data before too long as well. So it's just a big data time. So let me then go to metabolomics and proteins. So last Tuesday, we got our metabolomic data from these 41 samples. So what we did here was to take serum from each individual. We went with the company Metabolon, who looked at approximately 1,000 different metabolites. And when they compared the people with hEDS to the controls, they found that 104 had regulatory differences at a p less than 0.05 level. Another 75 were somewhat, questionable intermediate. So obviously in 1 week, haven't had a chance to give you every conclusion that we'll pull out of this. But at first glance, it looks like what's very striking and interesting about this are some differences in lipid metabolism. So here's the whisker plots, and words you might recognize and be able to read from a distance are endostrene diol, alpha-endostrene 3-beta, these and epiendosterone. Many of these are in androgen metabolism, and they're androgen metabolites. And so we're, in general, seeing a downregulation of androgens in people with hEDS. So bringing this back around then, when we think about steroidogenesis and the steroidogenic pathways, here are the androgens down here. And we're seeing that there's generally downregulation of these, which is somewhat the opposite of what you'd expect in conditions like congenital adrenal hyperplasia, where we have a block of CYP21A2 and then have an upregulation and an androgenization because nothing can go on to the right of the pathway, so everything to the left gets upregulated. So we're actually seeing the opposite of CAH in our population, which is kind of bizarre and not what you'd expect based on the population. But it's consistent with what those hints we were seeing in our early DNA data. We're seeing a lot of duplication of CYP21A2. We're seeing higher tenascin X levels in our participants, and we're seeing higher upstream androgens. It's an interesting hypothesis that we will absolutely be spending hours to try to work on further, but it might be the first glimpse at pathogenesis that we've come up with. So again, a slightly more readable set of all of these different androgen metabolites that are actually lower in our populations. So if those are lower, it means other things are higher. So again, we have to think of all the implications of this upstream and downstream, and we have to test this to make sure it's holding up. But it's an intriguing glimpse. So again, just to put this back into context, part of the reason it's so intriguing is CYP21A2 is right next to tenascin X, which is one of our favorite candidates, and we know it's key, a key step in androgen metabolism or in steroidogenesis metabolism. So the team that's done this work, again, I'll acknowledge them at the time. Sarah Hannon is my mass spec person. She's the one who is able to take the high impact data and pull it together. Maureen is working on TNXB. Zach is my local research coordinator who's helped with collection here. Karina was my research coordinator in Tucson who helped with collection there. And Jennifer is the PI in the Tucson group who's quite interested in data and data mining from medical records. And then Rohith is a hematologist down at St. Jude who's interested in the bleeding part of this work. So let me then summarize with this last couple pieces of data, which are the phenotype part of the data. So the question we're asking here is, we've taken what used to be one different diagnosis, EDS type 3, and split it into two diagnoses, hypermobile EDS and hypermobility spectrum disorder. And so as I said, we tried for our study in Tucson to collect only people with hEDS. But in the end, we ended up with a small set of people with hypermobility spectrum. And so we used the data from the objective findings on these individuals to do basically a logistic regression analysis and ask, are there any objective findings that allow us to figure out who has hEDS and who has HSD? Are there any of these crazy complex criteria that will help us to simplify this and say, go here, there's where the money is for making this diagnosis. And so what you see here are that a lot of zeros in this hEDS versus unaffected. When we looked and said what differentiates someone with hEDS from someone who's normal, who doesn't have syndrome, lots of these things differentiate. But in telling hEDS from HSD, only a couple. So unusual stretch marks, unexplained skin striae was one thing that had a p-value that was relevant, that was significant, and physiogenic papules, these little papules on the bottom of people's heels when they put pressure on their heels, where we say the skin splits and some sub-Q tissue sticks through. Those are the two things that differentiated. Not sure they were the two I expected, but they were what the stats showed. The third one that actually did was, that went to the wrong place, positive family history. The positive family history is circular. That's not a good argument. So basically, we're finding that the hEDS criteria, while developed for research and useful for research, not so good at differentiating these two old diagnoses. In addition, as I said in that original study, we did a bunch of questionnaires. We terrorized our participants and made them fill out 7 different long questionnaires about every detail of their clinical data. And so we know about kinesiophobia, fear of movement, fatigue, sleepiness, GI symptoms, autonomic symptoms, a variety of different somatic symptoms. And so we were able to ask, again, do the symptoms differentiate? Can we tell the difference who has which diagnosis based on their symptoms? Likewise, not really. If we take all of that symptom data and, again, run a PCA, thank goodness for Jennifer, because this is her baby, we came up with 3 groups, group cluster 1, cluster 2, and cluster 3, black, purple, and gray. And when we look at diagnoses, there's no clear differentiation between the 3 diagnoses. As a trend, cluster 1 includes most of our people who are unaffected, and cluster 3, but people with hEDS and HSD based on their symptoms don't differentiate. So our conclusion ultimately, when we reported these data in 2021, was the criteria are lovely, but they need some more work because they don't tell these 2 groups apart and they don't predict symptoms in any way. And really, we want to understand the etiology, but really what we want to understand is what symptoms people have and how to help them deal with them. So the symptoms are important. So deeper dive into that, really the only symptoms that varied between people with hEDS and HSD were some of the compass scales, some of the autonomic symptoms. So the secretomotor, the papillomotor, the vasomotor symptoms in people with HSD were statistically significantly different than in people with hEDS. And so that was kind of the only thing that differentiated. So we're again working to understand what this means, and thankfully as an international community there's working groups meeting every month at 7 a.m. on Thursday morning to sort through how to improve these criteria to make them more useful for physicians and for patients and for researchers. So that's okay, because I always talk too fast. At that point, let me summarize and say that, as we've pointed out, hEDS is complicated, understudied, and challenging. There are these new criteria that are useful, but perhaps not as useful as they could be. We still don't have clearly associated genes, although our group has some favorite candidates. There are some other groups that have some other candidates that we hope will be released soon. We're looking at RNA sequencing and metabolomic evaluations to help understand the relevance, and we're getting some intriguing things out of that, and are expecting a big bolus of data very soon. The integrin switch proposed by another group doesn't seem to be so relevant in situ. I'm becoming quite intrigued by these androgenic differences that could correlate with and or be directly related to TNXB and its pathogenesis. Obviously, I should, came up wrong. Thank mostly the patients who have been amazing and given literally their bodies for this work, and so at least live parts of their bodies, and also to our many, many donors. So, all right. Questions? Yeah.
Sorry, I came late, so maybe you have already mentioned. So why the in vitro studies didn't catch the in vivo, the skin, in terms of integrin switch? Is it because of the functionalization in vitro studies, or?
I don't fully understand why it didn't, but I do suspect it has to do with the very different environments in which skin in a tissue culture dish lives versus skin in situ. It would seem like there should be some relationship between them, but the other thing I would say, my Italian colleagues are lovely, but no one else has really reproduced that differentiation either. So whether this might be some unusual function of the way they froze or thawed or of their specific cells is still kind of up in the air as well. So it's a good question, and it felt like it should. So I'll be curious, though. The bigger, more important one is the reproducibility in our own tissue culture cells. If the cells we've cultured reproduce it, then we know that it's a difference in the in situ environment. If our cells don't reproduce it, then we know it's a difference in their lab. Okay.
So one is there any evidence that the actual rate of the disease is increasing, or are you just finding people more easily because people are kind of becoming more aware? And then the second part of that is, do you think, it's much more common in women, and a lot of things are, but women also have a lot more exposure to environmental toxins. And so has anybody done any work to look at how to link between environmental toxins that women are exposed to and EDS? I know, environmental toxins in mice, Johnny touched on causes fibrosis in uterus, and so there seems to be some things related to cause this.
I think they're both good questions that I don't have clean answers to. At least at first, I thought this was looking like the autism stories where autism was used to be diagnosed only in people very rarely, and now awareness is raised and we see a lot more people diagnosed, but the spectrum of severity has increased. So at first I thought maybe this is just an awareness thing, but I have to say over time I expected then we'd see a peak of people coming into clinic and then it would calm down, and we're definitely not peaked. So it makes me wonder if there aren't other environmental aspects. People haven't looked. I know there's overlap between communities. This summer I gave a talk at an environmental exposure group that's quite convinced that environmental exposures are important. But that's clinicians who are working and doing a lot with mold exposures, et cetera. So I don't think anyone has done a very high-quality study of this. There's some anecdotes that suggest perhaps, but there's I know of no clear data that are able to prove that. But I think I had an interesting discussion with the EIRH, and there could be some great things we could do with, for example, some of our samples. I believe we still have, aliquots of I know we still have aliquots of all of our serums. So they're still at -80 and they're still pretty fresh. So exactly, it really does feel that way. Every time you look, there's another experiment. And so Jennifer and I, and when we have our lab meetings, we really have to limit and say we can't. We're done. We've got our people. We've got our ideas. We've got too many directions, and you can't go off in all of them at once. But there's been a call. The South Carolina group has done very well with having, patient scientists working with them. There's also a lot more awareness, and so, as people get interested in pursuing projects and being the lead, there's definitely right directions to go. Yeah. I think age is important. I mean, what we know clinically is that in many ways EDS looks like early aging. So people with hypermobility are often diagnosed with wear and tear arthritis, osteoarthritis at a younger age. And actually, the little bit of funding the NIH has given to EDS has been through the Institute on Aging. So, but the fragility that the hypermobility leads to and the types of injuries seem to very much mimic aging. We know that the hormonal changes that happen with menopause also certainly influence some set of symptoms. And so it probably was a mistake to have quite this broad a set of participants. That was actually a bit of miscommunication between the two sites. We limited it at menopause. They kept going. So I made Tucson get all the controls to try to age match their 60-year-olds because I couldn't do it. But, so yes, age is important. It affects both the hormonal levels. It affects the symptom manifestations and what people are experiencing, and it seems to be often through collagen pathways. So yeah, that's got to be a variable that's taken into account. And in fact, when the metabolomics PCA worked out only by the age had to be added in as a variable for that PCA to look at all realistic. So clearly it's making a metabolic issue. Yeah.
I have two questions. First is, and I'm afraid I'm kind of novice, I'm an undergraduate, so I'm sorry.
No problem.
Very naive, but regarding the environmental/trauma impact in complement 4, you mentioned like the MNA section. You said that has been historically studied in schizophrenia and mental illness, and I was wondering if you had any more to say about how that might be related. And the other question that may or may not be related to the first question is that I saw that you mentioned it was really diverse team working on it, because it seems like you had a diverse group studying the disease. It looks like you were looking at, I saw women with fighting. So I was wondering if there's any, like going outside of that very general paradigm could also provide answers, especially as it's related to traumatic environmental impact and things that are more.
Those are actually both really good questions. So let me just address the second one, which is why do we have such a boring, ethnically boring group? And basically it's because we are just taking people who volunteer to come. So we really had hoped, in fact, in Tucson, which is about equal amounts Latino and Caucasian, to have a slightly more diverse group, but it just, the people who responded didn't. I think that part of what's going on there is not everyone, gets through the medical system up to the level of a subspecialist. And so there's clearly some discrimination happening at multiple levels within the medical system. There might be some recognition issues, but I have to say that my clinic reflects these numbers. I do see, patients of different ethnic groups, but the bulk of people I see would identify as Caucasian. The second question about the C4 and cognitive issues is interesting, and I have thoughts, but not so much data at this instant, which is when we got together as an international community in Italy this September, there was a whole session on questions of autism prevalence, especially autism prevalence and autism spectrum in EDS. And about half of the clinics seem to look like mine, and about half of the clinics look very different. In my clinic, we have a very gender diverse group, and we have a lot of people who are seeking and or who have been given diagnoses of autism and ADHD, a lot of people at various stages in the gender transition process who are exploring their gender. And what I'm learning is that gender questions and autism often go together and are often part of the same people. What we also know is that schizophrenia and autism are not at all the same disease, but they sometimes have the same risk factors. And so what I'm conflating in my mind is that perhaps some of what we're seeing are people who are having that the C4 variants might be a risk allele. That's not the right word for it, but might be changing the probability that someone is dealing with autism and or gender questions, and that might be part of this. The other way of looking at it, kind of the alternative way, is when I talk to my colleagues who care for patients with multiple sclerosis or with long COVID or with PTSD, a lot of the downstream symptoms that they're dealing with, a lot of the autonomic nervous system issues, a lot of the GI symptom issues are very similar. So part of what's also going on is, I think when the brain gets overloaded from whatever area, it only has so many ways that it can express that to the body. And so some of what we're dealing with, some of these symptoms, are just brain overload symptoms. And so we really should probably be having one functional clinic where all of our long COVIDers, a lot of our EDS patients, and a lot of our MS patients are all the same clinic, because it's the same issues. And why are we developing our treatments in silos? Because what they learn, I can learn from. So that's my opinion. That's my soapbox, and here I'm standing on it. Yeah. So I think there's both biological and biological genetic and biological brain issues why we're seeing some of the things we're seeing. All right, it looks like I've got you guys out of time. So thank you for your attention.